Hello, welcome everyone to the new episode of the Student Spotlight. It is our pleasure today to have one of our students from the Future Scholar Program, Kari Maxian. Kari is currently 15, going on 16. She's a high school student in upstate New York, and at the CCIR Future Scholar Program, she took on independent research under the mentorship of Dr. Struan Murray from Oxford. And she took a course titled Computational Genetics. So today we've invited Kari to share a little bit about her interests, her research, and her experience studying computational genetics at CCIR with Dr. Murray. So first of all, welcome, Kari.
Yeah, thanks for having me.
So Kari, I did a little introduction for you, but I think it'd be really helpful if you begin by sort of saying a little bit about yourself, what high school you go to, what your interests are, and tell us the title of your project and it's sort of a really brief abstract of what it was about.
Yeah, well, I'm Kari. I go to Manlius Pebble Hill School in Central New York. And I'd say I'm a pretty well-rounded student, but I definitely really like science, which is part of why I decided to take this course. And my project was exploring histone modification's role in epigenetics and gene expression. So histones are the proteins that DNA is wrapped around to package it tightly in the nucleus, and groups of 8 histones are called nucleosomes. And then so when the DNA is wrapped up like that, it can't be transcribed to make RNA to then make proteins. So when these nucleosomes are there, the promoter can't attach to the DNA to start transcribing it to begin the process of making proteins. So that's sort of how it comes in with gene expression and proteins and how things function. Because depending on the environment or certain things that happen, nucleosomes can move on the DNA to make genes exposed to transcribe them. So depending on how histones are modified, because the amino acid chain of the protein, different chemical reactions can happen to it, and that can either make the nucleosome more tightly wrapped, so it's harder for the promoter to access genes, or loosen it and make the nucleosome more free to move and make it easier for the promoter to transcribe the genes. So I sort of looked at how the histone modifications refers to the chemical reactions that can happen to the tails. So I looked at how some of the different reactions can either like increase transcription or reduce it, depending on whether it sort of opens or closes the DNA. And the 3 modifications I looked at, methylation, acetylation, and ubiquitination. I'm pretty sure most of those can go either way, making the DNA or the nucleosomes tighter or looser. But it was interesting in that the ones I looked at all seem to have a positive correlation with transcription. So they make the nucleosomes looser. And then I went and tried to find previous research that's been done on these. And I found that, yeah, I wasn't just like doing something wrong. These all pretty much do have a positive correlation with transcription. But obviously that's not the case for all of them. And there are like hundreds that I didn't look at that definitely do reduce transcription.
So those are a whole bunch of very big concepts. And I think it's going to be helpful for us and the listeners to sort of break things down a little bit. So could you begin sort of by telling us a little bit about epigenetics and what that is?
Yeah, epigenetics refers to sort of genetic information that's not really contained in the DNA. Like the histone proteins are not directly part of DNA or genes, but depending on how they're changed, they can make it so that more of a specific gene is transcribed and thus more of a certain protein is created. So it could end up making changes in the larger organism depending on how much of that specific protein is made. So then, like, if that organism had cancer and it changed how the histone modification worked and it made a nucleosome tighter when it should have been looser, so then now there's less of the protein that the organism needs. So now it's not going to function correctly. But it's not a problem with the DNA itself. It's something outside of it that is changing how the transcription works.
Could you sort of give a example of an easy to, I mean, the cancer example is helpful, but could you come up with some sort of more like simpler examples of what epigenetics would look like?
Well, there's, I believe, so the specific histones, there's certain like sort of terminology they're used. One specific histone modification is called H3K4ME3, and the H3 is the number histone of the octamer because the nucleosomes are a group of 8 histones, so it's the third one in that group. The K4, the K is lysine, which is one of the amino acids, so it's the fourth lysine, and then Me3 means trimethylation. So my chemistry needs to brush up on that, but that's like it refers to how many parts of it, or like 3 methyl groups are added, something like that, I think. But that specific modification, as part of epigenetics, that one is often seen as associated with higher transcription. So it when it's seen around like genes, it's associated with higher transcription. Like what I looked at, breaking down, because we got genetic data from all the genes in a yeast cell. So looking at the genes where transcription was lowest versus where it was highest, you could definitely see how the H3K4ME3 was associated with higher transcription. So when that modification was there, I believe, yeah, it's one of the ones that loosens the nucleosome. So when that one was there, it was making the gene easier to access and then easier for proteins to be created. So again, it's not something specifically in the DNA that's positive for the organism. It's something outside of that that is making it easier for that protein to be created.
So stepping back a little bit, so the specific course that you took with Dr. Murray is computational genetics. For those of us who are less familiar with that paradigm, from what I understand, computational genetics is kind of a new movement in contemporary biology that really exploded after we've developed the techniques to gather massive amounts of genetic data, and it's sort of a borderland interdisciplinary study that brings together data science and computational techniques together with biological data. So could you tell me a little bit about sort of the computational aspect of your research and sort of what that enabled you to see and what perhaps are some of the limitations of that approach that you came to observe.
Yeah, it really, computational genetics, like genetics itself is such a new field. So then combining the programming with it, like you said, it really is just something new and there's still a lot we don't know. Um, but what we did was we used MATLAB, which is a programming language used largely to manipulate matrices, which in our case were basically just data sets. Like you might have a chart full of stuff in Excel, and then you just bring it in here. And then MATLAB, wow, is incredibly powerful. And with just a line of code, you can do all sorts of manipulation that would be harder in stuff like Excel. And programming is not one of my favorite subjects, but it was really cool to be able to use MATLAB to do math, which I do like, and to explore science, which I also like, rather than just like, I don't know, programming a game, which is like, yeah, you do that all the time and it's not that fun, too many bugs. Not to say that there weren't bugs here, but it was easier in knowing that when I'm analyzing my data, you can tell right away when something's gone wrong, and it's fairly easy to fix it because it's mainly just syntax or the specific like symbols you're using. So what we did was in the first half of the course, we sort of learned the basics of MATLAB and the syntax and just sort of brushed up on programming logic. And then in the lectures, we were learning about the genetics and biology component. So then for the project, we combined those. So my data was all the histone modification stuff, the levels of those modifications for all the genes in the yeast. And then I used the programming language to take averages and make graphs and visualize it in a really cool way. And one thing which Dr. Murray had us do, which I'd never heard of before, but which ended up being one of my favorite parts of it, was so there were what, 5,000 or like 1,000 genes in the yeast data. And so looking at like the average gene length, and for each base pair on that average gene, having an average value of the modification levels, which he called a metagene. So it's sort of like your average gene, which was really cool to just sort of thousands of data values into just a simple curve where you could tell like, okay, here's where transcription starts, where the promoter starts to make RNA. And here's towards the end of the gene, and here's more towards the start. And that was really cool, being able to do that visualization with programming and just making it really easy.
Were there any sort of limitations of the computational approach that you observed as you pursued this project?
Yes, definitely. And I made sure to emphasize it a lot in my report. And Dr. Murray explained it a lot in the lectures too. The computational approach is really good for analyzing data once you have it. But since you're not doing like an actual experiment where you can isolate different variables and only change what you want, you can use statistics to find like correlations or stuff that's not related, or a bunch of different relationships, but you can't really identify what those are with certainty. Like it's definitely a correlation, not causation thing here, because without doing another experiment, you have no way to tell whether A is causing B or B is causing A, or they're both caused by something else that you haven't noticed. Because you're just looking at statistics and the data you already have.
So you mentioned earlier that one of the reasons you wanted to take this course was that you wanted to see if see what academic research is like and see whether it's for you. And I have a few questions about this for you. First of all, how would you compare your learning experience in this research-oriented course from, let's say, your biology courses in high school? What were some of the differences that you observed there?
Well, I mean, definitely, I guess sort of more hands-on in that we're working with like actual data. Um, I in my bio and chem courses, I think definitely in biology, we did a lot of experiments where we actually had our own data and were like drawing conclusions from it, like we were doing here. But in my chemistry, it was more sort of just seeing what like the reactions and stuff actually look like to give you a better idea of it. But this definitely felt like more of an independent sort of you're in control and you can look at what you want to look at instead of like, okay, here's your experiment and this is sort of what we want you to do with it. And you should probably be getting these results. Because Dr. Murray did give us sort of broad questions for each project choice to sort of investigate, but also gave us a lot of freedom and said, if you want to look at this, you can. If you want to go find some other data set and use that and look at that, you can do that too. So it was kind of cool to have that freedom in research that you don't really get besides choosing your like science fair project in middle school.
And how has this shaped your understanding of what research is, and maybe how has it shaped your understanding of what path you want to pursue in the future, if at all?
Well, I mean, it certainly hasn't narrowed it down for me at all in terms of what science I want to do because I find it all really interesting. Um, but I do have a sense um, that definitely academics is probably something I'd want to do more of because I found this fun and interesting. Because my mom is a doctor, a trauma surgeon. Um, and she's always told me, "Oh, don't go into medicine," especially as a woman. It's not fun. Um, but she actually used to be in academics, and I think she really enjoyed that. So I think that's definitely something I'd want to do more of, and maybe not as much the hands-on specific cases with patients, because I enjoy higher learning and getting to analyze data and do all this cool stuff, especially with all the new technology and advancements we have. It would be a shame not to go play with it.
And could you tell me a little bit about, so at CCR, for all our programs, we really like to emphasize the dimension of mentorship in all of our programs. And the way I like to understand that is that mentors aren't just teachers. They don't just transfer information to you. They really sort of guide you and try to help you out with your project. Could you tell me a little bit about your experience with Dr. Murray and sort of your experience in this course as you moved from the teaching components to the more research components? What was the mentorship dimension like?
Well, even from the beginning weeks, just in the lectures, it was really cool in that whenever we had questions, Dr. Murray was always really willing to answer them. And like you said, with mentorship, it wasn't just a one-dimensional, like, here's all the stuff you need to know, go use it. If we were confused or had like what if questions, he was able to address them. And sort of in a contemporary way, since he's active in this sort of new field. So we're getting answers from him that you might not be able to get from someone else because he's had hand-on experience and knows what he's talking about. And then he did also, once we started going into the research part of it, um, he sort of helped give us an idea of what we like, sort of general trends we should be finding, um, without being overbearing and saying, this is what you should be getting for each and every one of these histone modifications or, other genetic data. Um, but left it to ourselves while sort of guiding us and making sure we weren't going astray.
I want to now briefly turn back to your project and ask you sort of at a slightly more abstract level, what sort of, even though it's such a tiny little project that, when you were looking at the genetic data of yeast, but what do you think is the significance of this finding? What makes it interesting? And yeah.
Well, it definitely gives a good idea of like what we can do now, even just within computational genetics, because it does kind of feel like there was a lot I was able to find, and even a lot more I could have looked at, even just within this small little realm. And I mean, it can, since it's not humans, obviously you can't like get a direct sort of association, but it gives an idea of maybe more abstract general ideas of genetics that we could then apply to humans and, I don't know, cancer research and stuff with gene activity and things like that. Like, okay, in yeast, oh yeah, because that was one interesting thing I did find when I was comparing what I found with other research papers. Um, I think it was in plants. There were sort of a group of histone modifications that I think I don't, let's just say they were associated with higher transcription, but in humans or mammals or just yeast, they were associated with lower transcription. So it's interesting that some of these modifications, they across species boundaries, they share similar functions, but some of them are different too. And I obviously that's not as helpful for human research saying, oh, these actually aren't similar. But I think it's an important limitation to definitely think about um, with animal research and stuff like that.
And so you're currently a rising junior, is that right?
Yeah.
Yeah. And you're now sort of looking at college and sort of your um, how do you think this experience has helped you in thinking about sort of building your profile and on that sort of dimension?
Well, it's I haven't done a whole lot of stuff like this, but I definitely want to do more of it and maybe sort of solidify that science is what I want to go into. Um, yeah, and maybe seek out more things like this or just getting more involved with science, even if it's like volunteering stuff, because I really do enjoy it. And I think it's pretty clear that I don't know what specific science, but it's definitely going to be science. So doing as much of it as I can will probably help me decide.
I have sort of two more questions before we quickly wrap up. You say that initially you had come sort of your history teacher had pointed you to this program, and then you said something about how well that you found this particular course to sound really interesting. What was sort of the because as a whole, we do try to present courses that are a little bit special. So, you know, we, instead of doing like a biology 101 course or a zoology course, we try to do things like computational genetics, or in the upcoming round, we have things like quantum computing and quantum mechanics and particle physics. What was it like sort of diving into something a little bit more niche? And was that what sparked your interest, that pulled you away from the history teacher's suggestion?
Well, it actually ended up being like the sort of specialness aspect made it actually turn out really well for me. Um, because, yeah, what happened was my history teacher, it was just sort of a general thing. Like she posted on the Google Classroom for the class that, oh, here's an opportunity you might be interested in. And um, I did end up zeroing in on this specific science one, but then it ended up working out well because now I know more about genetics as sort of a foundation for if I take AP Bio or the school's genetics course, but also the programming aspect. I had been in a programming class, so it was sort of another way to branch out um, and make programming more fun for me. But also it was just really cool to see how it's not necessarily like a fringe subject, but it's pretty close in terms of like how new everything is. That was one of the coolest part, like learning all these techniques and terms and stuff that are really not that old at all, and just how much out there still is to explore.
And to wrap up, I'll ask the question I asked all the Spotlight scholars, and it's a very open-ended question. It's simply, is there anything else you wanted to share with the listeners, either about the course or about your experience or your mentor that I haven't yet touched upon and that you want to sort of close on?
Um, well, I guess what I would say is, I mean, I don't know about the other courses, but I would say, given that you're working with professors who are still very active in their fields, and at least with mine, it's a very new field, you're going to get the most out of it by asking questions and just not being afraid to ask questions if you don't understand, or also if you just want to know more, because these are the people who know what they're talking about and can definitely answer those for you.
Cool. That's some excellent advice for some future students. Well, I'd say that concludes a really interesting Spotlight Scholar interview. Thank you so much, Kari, for joining us, and best of luck in the upcoming school year.
Thank you. Thanks for having me. This was fun.